Welcome back, folks, to microbiology. Today we're going to be covering chapter 5 on microbial metabolism. So far in this class, we've talked about the history of microbiology, we've talked about the scientific method, we've talked about the microorganisms, some of the microorganisms that live in and on and around us, and we've also been introduced to some of the microbial agents that are important within pathology, even if they're not actually living systems. And we'll continue all of these discussions. We've also talked about the anatomy of a cell or a microorganism. And so today's discussion continues to kind of build on that anatomy and say, okay, now we know what the anatomy of a cell is. What is actually going on inside of the cell? Now, I'll tell you, spoiler alert, the amount of chemical and metabolic complexity within a cell is abundant. There's so many different processes that are happening within a cell. And so we won't cover all of those today, but what we'll do is preface the discussion and talk about some of the concepts and terms and systems and complexities and important agents behind microbial metabolism. And then in part 2, we'll go into specific examples of cellular respiration and fermentation and how those are occurring. So I invite you to go ahead and pause the video here, review previous content, preview chapter 5 and what we'll cover today, and then jump back in. So if we're to kind of put microbial metabolism or metabolism generally into a framework, we might start by asking what happens when we eat, right? Because anecdotally, we know that we need to eat to be able to have the energy and the nutrients for us to actually maintain our metabolic processes. Even on a macro scale, we'll start to get fatigued and we'll start to lose certain types of capacities to be able to grow and reproduce efficiently if we have short or prolonged hunger. And so when we eat, we're actually kind of coming back to some of those characteristics of life of being able to take in energy, nutrients, to maintain our living systems and stability and be able to grow and reproduce. And so it also allows us to think about, okay, when I eat, what is actually happening at the molecular level as I ingest, digest, and then actually those components of that food are spread throughout my body to all of the cells of my body via my blood and my circulatory system. What is actually happening when those molecules reach my cells? Why do they need to be there in the first place? Why are they so essential? And why do I keep on having to replenish that constant input of nutrients and energy, right? And so although our discussions throughout this course focus on microorganisms, the examples that we use in this specific chapter are actually relevant across many different types of organisms. So cellular respiration and fermentation occur in many types of organisms, not just microorganisms. They are kind of these general processes for the extraction and storing of energy, okay? Also, we actually utilize many of these metabolic processes in the kitchen. Sometimes we don't even think about them, but we're utilizing various types of cellular respiration and fermentation and various types of interactions with enzymes, for example, when we're cooking our food. And so there are a number of food science books that are very interesting that give you some insight into this chemistry. Dr. Harold McGee is one of the leading authors of food science work. And so I would recommend maybe looking into some food science connections as we're moving through this class. I'll provide some, but also think about the things that you're doing in your kitchen and how they're actually tied to these metabolic processes. So really, to preface and to start off this discussion, we need to chat about physics. And we need to connect that to biology. And so I want you to start to think about this discussion post, which is going to be the first part of your discussion for this chapter, and start to think about what are the laws of thermodynamics, the first and second laws of thermodynamics. What are the characteristics of living systems that we've talked about previously? And how do they interact with one another, right? So if we think about the laws of thermodynamics, we have the first law of thermodynamics, which is the conservation of energy, that energy is not created or destroyed within a system. It can be transferred from one form to another. Those transfers tend to be inefficient, and so there tends to be a loss of heat, but there can be a transfer of energy, but energy cannot be created or destroyed. So if we have living systems that need to be able to take in energy and metabolize and grow and reproduce, then they need to be able to find an energy source, and they need to be able to transfer it into useful kind of formats or forms, right? So if I go to a different country, I can't use American currency, for example. So the currency of the cell within energy systems has to be appropriate to it, right? The second law of thermodynamics is the law of entropy, that all systems are moving naturally towards a more stable state. But as we know anecdotally, more stable states tend to be more chaotic, right? So you have to put in a significant amount of energy in the morning to kind of collect yourself as an organized, kind of well-dressed individual, or you have to put in a significant amount of energy to organize your kitchen after trying to use it for cooking or whatever. You have to put a significant amount of energy into organizing your desk to be able to study, because those systems naturally fall apart. And the reason why they naturally fall apart is because it requires an input of energy for a system to be organized. Whereas if it is disorganized, it is more stable. If it's laying on the floor, it's more stable because it doesn't have anywhere else to go, right? And so within a living system, if we think back to the characteristics of life, living systems constantly must be organized. They must have organized complexity, and they must maintain homeostasis. And when they can no longer do that, they can no longer be living systems. They have to maintain that anatomy that we talked about in previous classes. They have to be able to maintain their metabolic processes to be able to grow and reproduce. Whenever they're not able to do that, then that living system will actually die. And so these concepts, you might think, well, physics, is this really connected to biology? But these concepts are integral to one another. Living systems are constantly moving against or moving within the laws of thermodynamics. And you'll see that as we move through this lecture. And actually, that first part of your discussion for this lecture is to make some of those connections between the laws of thermodynamics and the characteristics of life and living systems. So continue to take notes as we move through the lecture, and then you can kind of finish that up before you post it after we complete our lecture today. Okay. And so utilizing or thinking about those laws of thermodynamics, what you'll see oftentimes within living systems is that a living system's challenge is to take a reaction that might happen spontaneously, like a waterfall, but then break it down into smaller releases of energy, like a mill, and then couple that to some reaction or some way of conserving that energy so that it happens more efficiently, and so some of that energy can be conserved, right? And so this is essentially the processes that we talk about today, the enzymes that we talk about today, the different energy levels and energy states that electrons will move through that we talk about today, cellular respiration as a process as a whole. Generally speaking, this is what living systems are having to do, is take something that has energy within it and might combust or might fall like a waterfall, but being able to jump in there and step by step break down that release of energy and couple it with some other process that allows for the storage of energy or that allows for a specific metabolic process to occur simultaneously. And that's just what we do with dams and mills and things like that, kind of step by step, taking the energy from a system that has been built up to release energy, but step by step collecting that energy and then utilizing it for other systems. Okay. And so the aggregate of all of those processes that our cells are doing, no matter if they're, collecting energy or building something up or breaking something down, all of those processes collectively are called metabolism. Metabolism. And so metabolism allows for us to take the nutrients that we eat, we remember this guy here, take those nutrients that we eat, break them down, okay? And any process where you're breaking down components, that's called catabolism. And then using those units that we've actually derived from our food, build up our own systems. And that is called anabolism, when you add components together to build something up. And so all of the cell's metabolism is either breaking down components that are either not needed or they're needed in smaller units, or building up components so that we can grow and reproduce. Any system where we're breaking down components, so any catabolism, releases energy. And anytime you're building up components, that requires energy. And we know this anecdotally. If we were to push over an old wall of a building, it would just kind of spontaneously fall and release energy. We would hear sounds, we would see dust, maybe the ground would shake a little bit. That's all releases of energy. But if we were to build up a wall to actually build a building, you would need folks, you would need materials, you would need energy being put in to actually structuring and organizing that wall. And so that would be anabolism, adding things together, and that would require energy. And we're going to look more at those a little bit more specifically in a bit. But before we start to break down these concepts into simple forms, I first want you to look at this. Okay. The metabolic metro map. And I don't put this up here because I actually want you to see all of the details within this diagram. I put this up here because I want to remind us that we are going to break down some of these components and look at specific steps within some of these pathways. But really, each step is part of a whole integrated system of different types of chemical reactions, of different pathways, of different feedback loops that sometimes will turn a system on or turn another process off. And they're all integrated within the cell or within different organisms within a community, right? So when one organism eats another organism so that it can have energy and nutrients, or when an autotroph, for example, does photosynthesis that actually provides energy to the community, you're working within a complex system of metabolic processes that all come together so those organisms or those communities can function. And so we won't be able to look at all of this complexity, but we will start to understand some of the important foundational laws or the important foundational structures or the important foundational concepts within this complexity, right? And so kind of keep that in the back of your mind, and we'll come back to it at the end of our lecture today. But we will start to break down some of these ideas and concepts, all the different metabolic processes that we need within our cells and within our systems. And so much of it we're still learning about and we're still understanding, right? Oftentimes, the two really foundational processes that allow us to understand some of these metabolic systems and some of these food feedback loops are the processes of photosynthesis, which is a process that builds up the energy that's coming into the plant, oftentimes, or the photosynthetic organism from the sun, and then the carbon that it's taking from the atmosphere and builds up those carbon-carbon molecules using that energy from the sun. And so what is that? Is that a catabolic process or an anabolic process? Photosynthesis. When we're building up a glucose molecule starting off with carbon just from the atmosphere, but then building it up using the energy from the sun. Right? So if you said an anabolic process where you're building up or adding components together, which is a process that requires energy, then that is correct. Photosynthesis is an anabolic process. The flip side of that is, okay, the plant has glucose or the bacteria has glucose. Now what is it going to do? Is the glucose the functional unit of energy? No, it's not, which we'll learn about in this lecture. Glucose is not the functional unit of energy. So we need to now take that glucose and step by step by step, release the energy that was built up into it in its chemical bonds, and then produce usable forms of energy that can be actually circulated throughout the cell and utilized within the cell, right? And so these tend to be similar opposites when we look at metabolic processes. So question, is respiration a catabolic or anabolic process where we take glucose and stepwise break it down just like that mill so that we can release energy in a form that is actually useful to the cell? So hopefully you said that it's a catabolic process. It releases energy and breaks down chemical components, okay? And so these two processes really give us a good grasp of what's going on with both anabolic and catabolic processes. They are also the foci of your chapter. We actually will focus only on respiration and its parallel but abbreviated sister, which is called fermentation. And we will not focus on or cover photosynthesis. I assume that you've covered that in prerequisite classes, and so we will focus on respiration, becomes essential to many of our discussions in this class, and so therefore that's going to be our focus for this chapter. Okay, so I do invite you to read those portions of the text on photosynthesis. They're very useful in understanding these concepts, but our focus here will be respiration. Okay. Now, no matter what we're talking about, photosynthesis, respiration, anytime we're talking about metabolism within the cell or metabolic processes or metabolic pathways, there are a couple of kind of key concepts that we're working, okay? And these go back to the discussion that we had on physics and biology, essentially. And so the first is that every cell acquires nutrients. It must acquire nutrients. It has to have the components to actually build itself up. Along with those nutrients, you need some source of energy to actually be able to utilize those nutrients. Nutrients without energy really don't give you the capacity to do anything with those nutrients. So you need some sort of energy source. Oftentimes, the energy source at the base of the community, the group of organisms, not just an individual organism, but the community of organisms is light. And so your photosynthetic organisms, be they plant or a bacteria that is photosynthetic, tend to be our source, our initial source of that conversion of light energy into a chemical form or chemical bonds that we can then actually utilize thereafter. There are some exceptions. So like some extreme environments or hydrothermal vents, the energy source is heat or chemicals instead of light. But most systems on Earth, the initial source is light. We also need a currency, a way of storing that energy that is consistent. And so what you'll see throughout biology and microbiology and living systems is that although there's a lot of diversity, that diversity sits on similar standard concepts. So all living cells, they have the same currency of energy, which is a molecule called adenosine triphosphate, right? So they take, organic components maybe that they consume if they're heterotroph, and then break those components apart and make ATP, or adenosine triphosphate. And ATP, or adenosine triphosphate, is what actually circulates the cell and is used to perform the various work or various pathways that are within that cell, or within those metabolic processes. So there's a standard of energy currency throughout living systems, and that is adenosine triphosphate. Okay. Also, cells catabolize nutrients to form precursor metabolites. So we have within our metabolic processes many metabolic kind of pathways. So we go back to this image here, many metabolic pathways. And the pathways actually feed into one another. And this is a really essential concept. The pathways will actually feed into another. One pathway will produce a product. When that product is made, maybe it'll feed into another product that allows a process or metabolic pathway that allows you to break down that product. If that product is not there, that secondary metabolic pathway won't occur, right? And this is also important on the community level. That precursor metabolites, oh wait, right here. Precursor metabolites sometimes are made by cells within a community and picked up by another organism. And so symbiosis actually builds on this same concept. So within our gut, for example, the microorganisms in our microbiome, they break down materials that we maybe couldn't necessarily break down, release those metabolites into our gut, and then we can pick them up and utilize them in our metabolic processes, right? And so this, not only within the cell, but within communities, is a way of kind of stepwise breaking down nutrients or systems that have energy within them, and then sharing those nutrients and energy within the cell or between cells in a community. Precursor metabolites, energy from ATP, and enzymes are all used in anabolic reactions, okay? And so we take all of these different components, energy, currency, which we said was ATP, the precursor metabolites, which are being produced off of other metabolic processes, sometimes other organisms, and then enzymes. And this is something we're going to talk about more, but these are essentially the structural components that allow us to do various chemical reactions. They're the structural components that hold the substrates together, or bring the substrates together, or activate the substrates so that you can actually have not only anabolic reactions, but also catabolic reactions. Enzymes are the workers of the cell. For us to actually build up our systems and our macromolecules, we need enzymes, which are the workers, and energy, which is in the form of ATP, okay? And that is how we, as cells or organisms, grow, is by assembling macromolecules from the precursor components that come in via the food that we eat in our nutrients, and the energy that's been transformed into ATP. Okay? And that's also how we reproduce, because we will need to be able to grow and then move that genetic information on to offspring. Now, at a bacterial level, it's very simple. The cell just grows until it's almost doubled in size and it splits. If you're talking about macroorganisms and sexual reproduction, that's a little bit more complex. But if we're just talking about a bacterial cell, they're just taking in the nutrients, the precursor molecules and the nutrients. They're using the ATP that's been transformed into that currency of ATP, and they're growing, doubling in size, and then asexually splitting, right? And so these concepts kind of guide our thought process and our discussion today as we talk about metabolic pathways within microbial agents and microorganisms, within microorganisms specifically. And here's an overview of that.
As organisms live and grow, they're constantly in the process of making and breaking bonds in molecules. Metabolism is the sum of all the chemical reactions that take place in an organism. Metabolism includes catabolism and anabolism. Catabolism includes the processes that break down complex molecules into simpler molecules while harvesting their energy and storing it, usually in the form of ATP. Anabolism includes the processes that build more complex molecules from simpler molecules. The energy acquired through catabolic processes is used to drive anabolic processes. Neither catabolism nor anabolism is completely efficient, so at each step, some of the available energy is lost into the environment as heat.
Okay. And so as we move through, we're going to talk about some of these different types of pathways, and then we'll talk specifically about cellular respiration. Okay, so we've already introduced the idea of catabolic and anabolic pathways. So our catabolic pathways are the pathways that allow us to take usually nutrients or food that we're ingesting and break it down into components that can then be built up or synthesized. And then that's kind of a cycle, right? And that can be a cycle within the community. So I might have a photosynthetic organism like a pea plant that builds up glucose within its structure. And then me as a heterotrophic organism, I might ingest some of those components and then break them down in a series of catabolic reactions that then allow me to build up my structures in a series of anabolic reactions, right? Or it could be happening within the same cell where you have a photosynthetic bacteria that is producing glucose and then breaking it down to release the energy from that glucose molecule, producing glucose through photosynthesis, which is an anabolic process, and then breaking down that glucose through catabolic processes to then generate ATP, right? And so however that is happening and whatever complexity is happening around, catabolic processes release energy, so exergonic exit of energy, and anabolic processes require energy, so ender or input of gonic, which is energy. And so we can see those processes here, endergonic reactions or your endergonic reactions or anabolic processes are uphill reactions. So you need to put in energy to get to the products, whereas your exergonic reactions are downhill reactions, catabolic or exergonic reactions are downhill reactions, and energy is released. Okay. You'll notice that each of these requires a little bit of activation. So even though, like if we think of a downhill reaction, like a ball rolling down a hill, it requires a little bit of a push. And here, endergonic reactions require a lot of energy input. So keep that in mind, because we're going to get back to that in a second. That is called the activation energy that's required to start a process, even if it's a downhill process, even if it's kind of a spontaneous downhill or exergonic process within catabolism. It still requires a little bit of an initial push. We call that the activation energy, and that becomes really essential within living systems. So just kind of hold that in your mind because it's related to these graphs, and we're going to get back to that when we talk about enzymes. Okay. And so within the cell, we're going to be doing both of these processes. We're going to be breaking down, larger molecules and then building them back up into macromolecules through various processes. Okay. Now, a lot of the way that the transfer of energy is driven in the cell is based on what we call oxidation reduction reactions. And this is the transfer of electrons. And actually, we already know this anecdotally because electricity is essentially the transfer of electrons within metals that are conductors, that are insulated by insulators, which don't transfer electrons, and those electrons just move through those wires, and that's how we get the activation of, whatever electronic that you have around you, is through the flow of energy within these electric kind of electric wiring. And that's because the electrons themselves are moving through the molecules within the wiring, or moving through the atoms within the wiring. And so you have this movement of electrons just like a bucket brigade, and that's how the energy is flowing. Okay, so that's kind of at the macro scale. The same thing is happening within the cell, is that we're making and breaking molecules oftentimes by coupling that with the movement of electrons from higher energy states to lower energy states, back to higher energy states, back to lower energy states. And that coupling, or that movement of electrons either within an atom or between atoms, is what allows us to move energy between some of the different molecules that we're working with. Okay, and so we have molecules that allow us specifically to transfer electrons. And then those molecules also allow for hydrogen, which is then useful in creating hydrogen pumps so that we can make ATP. And we'll talk a little bit more about that. But we have NAD+, which becomes NADH. And so it can transfer, there's a redox reaction, transferring electrons. We have NADP+, which becomes NADPH, and we have FAD, which becomes FADH2, okay? And these molecules allow us to move electrons around so that we can get our end product, which is ATP, or adenosine triphosphate, which is really our goal in terms of the energy currency of the cell, okay? When we move electrons around between one molecule to the other, it is that outer electron, what we call the valence electron, and certain molecules are more likely to transfer electrons than others, or certain atoms are more likely to transfer electrons than others. This is one of the reasons that oxygen is both essential and important, but also dangerous within cells, is because oxygen tends to transfer electrons. And so we'll talk more about that when we get to free radicals. But essentially, when an electron is gained, then that molecule is the acceptor of the electron, and it also is what we call reduced. Now, that initially is like not intuitive. Why would it be reduced if it's gained an electron? So think about that for a second. Look at the diagram. See if you can understand why we would call this atom or molecule reduced if it just gained an electron. Okay, so you might know, or you might see that the electron is negatively charged. So when we move an electron onto an atom or a molecule, that actually reduces its charge. It makes it more negative. And so in that way, we're actually getting a reduced molecule in that case. And then the atom or molecule that loses the electron is called oxidized. It's called oxidized. And so some folks will use the term oil rig. Oil rig means oxidized is lost, reduced is gained. Okay. Oxidized is lost, reduced is gained. You can put that in here. Maybe add a text box where I say oil rig. Okay. Oxidize is lost, reduced is gained. Okay. And so you can see that here.
Many metabolic reactions involve the transfer of electrons from one molecule to another. This transfer of electrons involves two coupled reactions, oxidation and reduction. These are often called redox reactions. Cells use redox reactions to transfer electrons from one molecule to another within or between metabolic pathways. such as the Krebs cycle and electron transport chain. In an oxidation reaction, a molecule called the electron donor gives up one or more electrons to another molecule called the electron acceptor. In the oxidation reaction, the donor molecule becomes oxidized. A reduction reaction occurs when a molecule receives electrons from another molecule. The electron receptor therefore becomes reduced. The term reduced refers to the receptor's electrical charge. Because it's gained a negatively charged electron, the overall charge on this molecule becomes more negative and is said to be reduced. You can now see that the oxidation reaction must be coupled with a reduction reaction. In other words, if a molecule is to donate an electron and become oxidized, then another molecule must accept that electron and become reduced.
Okay. So we said that we use this kind of bucket brigade method for transferring electrons from higher energy states to lower energy states. And we use NADH, NADPH, FADH2, all of these to transfer that energy. But our end goal is to make what? Is adenosine triphosphate, ATP. So let's talk a little bit about adenosine triphosphate and why we even want that, or why it's so useful as the currency of a cell. Why don't we just use NADH? Why don't we just use NADH2? Why don't we just use, NADPH? Why don't we just use FADH2? Okay, so let's look at adenosine triphosphate. Okay. So adenosine triphosphate essentially has this amine group here, of this adenosine group here, and then it has 3 phosphate groups. So a phosphate is a phosphorus with oxygens attached to it. And essentially, within these bonds between the 3 phosphate groups, there's a significant amount of energy that is held. Any bond, any chemical bond is going to hold some sort of amount of energy in it, right? The more stable it is, the more energy it tends to hold. But in this case, we're actually able to pop those phosphates off because of their chemistry and release that energy, significant amount of energy, but then easily released. And then when we do that, we can couple other reactions with that release of energy. So as the energy is released, we tie it to a specific reaction that we need done in the cell. And so that allows for us to kind of do various reactions within the cell. And so what we do is our cells, bacterial cells, whatever it is, they actually utilize through the making and breaking of nutrients that come into the cell, coupled with the energy that is kind of released from or held within those molecules. Then eventually they make ATP, and then that ATP is what's circulated throughout the cell. ATP, not only can you easily access the energy off of it by making and breaking these bonds between the phosphates, but it's also highly water soluble, and it can be used in other ways. It can take on different functions within the cell. And so it is a really, really beneficial and useful molecule. And so here you can see you have, again, that nitrogen group here, and then the 3 phosphate groups that are attached here. This is just the same molecule, but shown just a little bit more clearly. And then each of these bonds holds the energy within it, but it can also be broken. And when that bond is broken, then energy is released. And so what we do in the cell is we cycle between adenosine triphosphate and adenosine diphosphate. So between an adenosine that has 3 phosphate groups attached and one that only has 2 phosphate groups attached. And then those are recycled within the cell. So you don't lose this molecule every time you use it. You just, those components, the adenosine diphosphate with the 2 phosphates and this inorganic phosphate that's just kind of free floating within the cell, then are remarried or reconnected to make adenosine triphosphate, which can be utilized again. And that's why you have to keep on eating, is you have to keep on supplying the nutrients and the energy that you need to keep on recycling ADP, adenosine diphosphate, back into ATP, adenosine triphosphate, so your cell can keep on doing these reactions. There's a couple of ways of doing that, a couple of ways of recycling ATP. You could have phosphate level phosphorylation. Phosphorylation is just reconnecting the phosphate to the ADP, the adenosine diphosphate, where you literally have a phosphate on one molecule that pops off and then is attached to the adenosine diphosphate. That happens, and it will occur in the beginning steps of cellular respiration. We'll see that there. But usually the way that these phosphates are recycled is through oxidative phosphorylation, which is where you actually use a flow of electrons and hydrogen ions to generate more ATP. So that flow just takes the inorganic phosphates that's in the cell and then allows you to connect them via an enzyme called ATP synthase, or an enzyme that synthesizes ATP. You can see from the name, right? You could also have photophosphorylation, where the energy from the sun or from a light source would actually be able to energize this reaction. Also less common, but does occur in some organisms. Okay. But essentially what we need to do is find a way to generate ATP and have a consistent supply of ATP within our cells. And we know this anecdotally, that if we don't eat, then we start to feel fatigued. And that's essentially at this ATP level. You're not recycling your ATP. You don't have enough energy currency in your cells, and you start to feel fatigued. Prolonged fatigue will actually lead to the cell itself not being able to do its metabolic processes, not be able to grow and reproduce, so on and so forth. So that becomes a really big issue if it's prolonged. Okay. Now, we said that the energy source for cells is ATP, but who are the workers? Who is actually going in and using that energy source and connecting or building up the macromolecules, or breaking down the macromolecules to release the energy to then rephosphorylate ATP? Well, we had mentioned them before, but these are called enzymes. Enzymes tend to be proteins. So if you remember back to your review of macromolecules, and you're always welcome to pause the video and review macromolecules, proteins tend to be the macromolecule category that makes up enzymes, although there are some enzymes that are really essential that are also made up of either full or partial nucleic acids. Okay, like ribosomes. Ribosomes are actually a combination of proteins and nucleic acids. And so these enzymes are biological catalysts, and they are the ones, if you remember, I told you to hold this in your mind. This little bump where any reaction needs a little bit of a push. Even if it's a spontaneous reaction, like a ball rolling down a hill, it needs a little bit of a push. And if it's not a spontaneous reaction, it needs a lot of a push. Enzymes are the ones doing this push. Okay, within a living cell, you cannot just hang around and wait for the ball to roll down the hill. If that's a reaction that you need, you cannot just hang around and wait for the components that you need to build together to run into each other and spontaneously make a wall. That won't happen because it needs energy to be put into that system. So the workers of the cell are enzymes. They are the ones that actually make reactions more energetically favorable by decreasing what we call the activation energy, which basically just means giving whatever reaction the push that it needs to happen. What is the push that it needs to happen within a cell? Well, you have different substrates, different reactants that need to come together for that reaction to occur. The enzymes will bring those reactants together. You need to energize that process so that that reaction can occur. The enzymes will energize the process. You need to bend a bond a little bit so that it will break within a molecule. Enzymes will do that. And enzymes are very, very, very specific to the specific task that they have. And that is because their structure is what allows them to do their task. And they have a very specific structure. that's coupled with that task. And we're going to talk more about that. But first, let's look at an overview of enzymes.
Imagine a chemical reaction with the general form AB yields A plus B. In order for this reaction to occur, the reactant molecule, AB, must acquire enough energy to disrupt its stable electron configuration so that its electrons can rearrange to form A and B. This energy barrier is called the activation energy of the reaction. In living cells, enzymes serve as biological catalysts, speeding up reactions. Enzymes accomplish this task by lowering the activation energy of the reaction, making it easier for reactants to acquire the activation energy required to produce products.
Okay. So, just a fun little question. If we were doing a reaction that is AB goes to two separate components, A plus B, is that a catabolic reaction or anabolic reaction? And so hopefully you said catabolic reaction because it's a reaction that breaks apart components instead of adding them together. An anabolic reaction would add components together. Now, then the next question would be, is this an endergonic reaction or an exergonic reaction? When you have a catabolic process that's breaking components apart, is that endergonic, where energy is input into the system, or exergonic, where energy is released from the system? And you can see that from the diagram here. So what would you say? So you can see that energy is being released from the system because the reactants have more energy in them than the products do. Okay, so it's an exergonic reaction. Okay, but even though it's an exergonic reaction, it needs a little bit of a push. It will happen spontaneously once it gets started, but it needs a little bit of a push. And the enzymes are what give it that push by decreasing the activation energy, bringing the components together, stressing the bonds a little bit, and allowing for the reaction to occur. Okay. There are a number of different types of enzymes within our cells, from hydrolases, isomerases, lyases, transferases, and you can even tell from their name what they're doing sometimes. Transferases, moving functional groups from one molecule to another. Oxidoreductases, right? Moving electrons from one molecule or one atom to another. Lyases, lyse or lysing is to split or to break apart. Hydrolases, so hydro in relation to the movement of water molecules, right? And so many different types of enzymes within our cells, and each enzyme, even within those categories, has a very specific function. So usually a reaction is actually not happening because of one enzymatic, a single enzymatic activity. It's actually a series of enzymes that process the metabolites from the previous enzymes. And that is actually beautiful because it allows for feedback loops. So you might have what we call a metabolic pathway, which is a series of enzymes working together, and they actually respond to one another. So if you have too much of the end product, then that end product will turn off that pathway and redirect some of the intermediate products into other pathways. So remember that really complex diagram. All of these reactions are connected to one another, and oftentimes they're happening because of multiple enzymatic activities, sometimes hundreds of enzymes being involved in a process like cellular respiration. And those enzymes make different intermediates that then interact with one another, and we'll talk more about that in a minute as well. Also, enzymes oftentimes can be kind of improved, their activity can be improved or augmented by cofactors and vitamins, very essential to make sure that they're functioning correctly. And they have very specific shapes. So we said that each enzyme is performing a very specific function based on its very specific shape. And don't think of shape as just the physical shape. Also think about the the charge shape. So different types of hydrogen bonds, because of different polarities within a molecule, tend to be really essential to how molecules interact with enzymes. So think about both the physical shape, but also the little magnetic forces or little charges within the enzyme, as well as being the way that it interacts with its substrates. Speaking of interactions, the way that enzymes interact with their substrate is called induced fit. Okay. And oftentimes what your textbook will show you is like a very specifically shaped substrate that kind of, connects with a very specific lock and key type format of an enzyme. But like we said, it's not just the shape, it's also the interactions. And that induced fit means that when the substrate comes into the enzyme will actually change shape to hold on to that substrate and interact with it and either build it up or break it down or energize it or whatever it is. And so I don't love the key lock and key kind of model of induced fit. Really, it's much more interactive, right? So you have here Mr. Stark, you imagine him as the substrate, and then his suit as the enzyme is actually fitting to the substrate. It's interacting with the substrate, and then it's whatever, energizing the substrate or pulling it together or whatever it is that it's going to do then. So induced fit is much more dynamic than just the lock and key, very static kind of example that we're often given. And so then that enzyme can then act upon the substrate. In this case, energizing or strengthening Mr. Stark here. Okay. And so you can see here an example of an enzyme that is actually part of the enzymatic pathway within cellular respiration, taking a 6-carbon molecule here and then breaking it down into 2 3-carbon molecules. And that breaking of the carbon-carbon bonds releases some energy that can be coupled with the transfer of electrons that can later be coupled with the production of ATP. And we're going to go through that step by step in the second part of this video. Okay. So here, this video is going to give you an idea of some of the steps within reactions that gives you an idea of how enzymes work. Again, remember that it's much more dynamic than we can really show you with these very simple diagrams, but it gives you an idea of how enzymes are working.
Although every enzyme performs a different reaction on different substrates, they all go through the same general steps in accomplishing their reaction. These general steps include substrate binding, formation of the enzyme substrate complex, product formation and dissociation, and enzyme recovery. Like all catalysts, enzymes are unchanged by the overall reaction. Now let's look at each step in more detail. The first step in any enzymatic reaction is the binding of the substrate by the enzyme. The enzyme includes an active site that can accommodate the size, shape, and electron configuration of its substrate or substrates. According to the induced fit model of enzyme-substrate interaction, a perfect fit does not form until the enzyme and substrate bind to form an enzyme-substrate complex. After the substrate is bound in the active site, the enzyme-substrate complex is formed. The shape of the enzyme may change to allow for an efficient chemical reaction, and amino acids may accept or donate protons or electrons to facilitate the chemical reaction. Once the enzyme-substrate complex is formed, and any reactive groups have moved to the right position, the reaction proceeds and products are formed. The products dissociate rapidly, leaving an empty active site. After the products leave the active site, the enzyme returns to its original configuration. Any groups that donated or accepted protons or electrons are also returned to their original state. Like all catalysts, the enzyme is unchanged by the overall reaction.
Okay. So we've talked about how that shape of the enzyme is really, really essential, right? And if you remember back to proteins, proteins have a kind of like a linear composition of different amino acids. Like, okay, this is the series of amino acids, like beads on a string or like beads on a necklace. But that string of amino acids is not functional until it's actually folded in a very specific way, right? And so that means that any environmental factors that impact that folding, and a folding tends to be based on hydrogen bonding, so different polarities and partial charges within the molecule, anything that disrupts that or anything that inhibits that or anything that improves those interactions is going to impact that enzyme. And so if I'm sure you've talked about this in prereq classes, but also it's an important way that we interact with our food. We're going to talk about that in a minute. But anytime you have environmental factors that influence the shape of the enzyme, it is either going to be improved in its function sometimes, but oftentimes what happens is it denatures. And denatures means that it unfolds and then it doesn't have the capacity to do its work anymore. And so kind of the one exception to that is temperature. Initially, as you increase the temperature of an enzyme, it actually improves its function. But then it gets to a point where it immediately essentially starts to degrade because of that increased activity, degrade the structural components of that enzyme, and then it's denatured and it's no longer functional. Okay. And so this is like our slow cooker example. As you slowly increase the temperature, actually the enzymes in the meat itself will have some function. But then at one point it'll get so hot that those enzymes will degrade. And that's when you get like the thickening or hardening of the meat. And it changes its texture from that raw meat texture to cooked meat texture. Okay. So temperature with things, environmental factors like pH, the pH has to be very specific to the enzyme. If it's an enzyme that's functional in, outside of the stomach in the human body, then it needs to be about a neutral pH. If you're talking about an extremophile, bacteria that's an extremophile and lives in very acidic or very alkaline conditions, then those enzymes work within those specific pH conditions of pH, right? And so it has to be within that range. If the cell itself or the environment where that enzyme is working becomes outside of that pH range, either higher or lower, then those enzymes will denature and they won't be able to perform their function anymore. Some other examples of factors that will impact enzymatic activity is, for example, the saturation of the substrate. So at one point, you have so much substrate or the reactant that's being used in that reaction, it'll basically plateau off the activity of those enzymes. Something that's really important to note is if we have the optimal conditions, temperature, pH, and then other conditions within the cell, then those enzymes will continue, they will not denature, and they will continue to be recycled. And they will not be used up in the reactions that they actually induce or facilitate. But if they are denatured, then oftentimes you have to have new enzymes that are produced. There are some chaperone proteins that can help refold enzymes and proteins if the change or the misfolding is not extreme. But if the environmental conditions really denature a protein completely, oftentimes you need to make a new one and fold it correctly before it can be utilized. So you can imagine if many different enzymes are being denatured within a cell, if that happens consistently throughout the cell, then the cell may not be able to maintain its metabolic functions anymore and may die. Okay. This is just an example of a functional protein and how it's folded in a very complex way. And then this is a denatured protein. There's actually a video on how we utilize temperature, acids, and some other factors in the kitchen that has some really nice kind of 3D images of proteins and what happens to them under high temperature conditions or acidic conditions. So I'm going to direct you to the food chemistry video that's in the description. Have you watch that and then come back into the lecture here. Okay, welcome back. So let's look a little bit more into, sorry, that's the discussion that we'll get to in a minute, but I want to look more into enzymes. So enzymes, they're able to do these various functions, but your cell also needs to be able to control these enzymes. And so there are a number of ways that a bacterial cell or a human cell, whatever it is, will actually control the activity of enzymes. And so you could have, essentially, there are various feedback loops. Some feedback loops will activate an enzyme, some feedback loops will inhibit an enzyme, and that activation can happen in the active site of the enzyme, where the enzyme is actually interacting with the substrate itself, or in a separate site called an allosteric site. Okay, and so let's look at some of those examples. We'll start with an allosteric activator. So here is a separate site that's different from the active site of the enzyme, right? So that's the active site. This is the allosteric site. But when that activator is there, then it changes the shape of the active site and it allows it to actually do its work, right? You might also have an inhibitor or an activator. Usually it's an inhibitor that comes in directly to the active site. And it has a similar shape to the substrate and will come in and actually block activity. And so that's how cells will sometimes turn off pathways, is that they have these inhibitors that can block the activity of an enzyme. Some toxins actually work this way too. So you have the toxin comes in and blocks an essential metabolic pathway, and then that's how toxicity occurs, okay? You could also have inhibition from an allosteric site. So a molecule that comes in and turns off the enzyme using a different site from the active site. And so that's kind of different examples of how a cell might turn on or off an enzyme. And usually that happens within a metabolic process where, or a metabolic pathway, where you have multiple enzymes that are all making intermediate metabolites. And whatever is the end product, for example, might actually turn off an earlier enzyme via an allosteric site or an active site inhibition, right? And then maybe when that end product diminishes, the quantities of it or the concentrations of it diminishes within the cell, and then that one molecule is needed, it pops out of the allosteric site and it turns the pathway back on. And so oftentimes you have these feedback loops that allow for you to not only allow for the cell, whatever cell it is, a bacterial cell or a eukaryotic cell, to essentially control the activity of not just one enzyme, but a whole pathway of enzymes. And these pathways are connected to one another via oftentimes intermediate metabolites, okay? And we're familiar with some similar feedback loops, like with a thermostat, right? Your AC is maintained at a specific temperature, but if somebody opens a door or window and it becomes too warm above that temperature that's being maintained, then the system kicks back on and actually cools the house again or warms the house again back to that temperature and then turns back off, right? And so feedback loops in our life, similar to how our cells or bacterial cells are using feedback loops to control enzymes. And so that concludes our kind of introductory discussion about microbial metabolism. That's part one. We now have part two, where we're going to talk more specifically about cellular respiration. Okay. And when we talk about cellular respiration, it'll give us an example of a number of these concepts. So if we look back to those 8 like key concepts that allow us to understand metabolic reactions, every cell acquires nutrients, metabolism is required. Metabolism requires energy. Energy is stored in ATP. Cells catabolize nutrients to form precursor metabolites that are then used. These precursor metabolites plus energy plus enzymes are what actually are doing the reactions. Enzymes in cohort with ATP are able to build up molecules or macromolecules. And cells grow by assembling macromolecules, and cells reproduce by doubling in size. So these kind of concepts we'll be able to see them in cellular respiration. How do we take in nutrients, break down those components, couple them with the movement of electrons, then actually make ATP that can then be used within the cell to do various reactions, both anabolic and catabolic, that allow for the cell to maintain its homeostasis, to grow, and to reproduce, right? And so although it's not a complete discussion of metabolism, cellular respiration really helps us to understand. And so I want you to think about, as you're moving between the parts, think again about this discussion of, let me get rid of this prompt because we don't need that part. Think again about the laws of thermodynamics and the characteristics of life and how they're tied into these concepts that we're working with in this class. And also, I'll invite you to watch either the less detailed or more detailed review of cellular respiration that is linked in the description below so that you can kind of jog your memory as to what cellular respiration is. And then we're going to get into, in part 2, kind of the steps of cellular respiration as well. Okay, so thanks for listening, and I'll see you in part 2 of chapter 5 in a minute. Take care.